So it's Oxford-style debating here in the U.S. Any of the very hot topic issues will be chosen on a week or every other week basis. And then usually, I think it's housed out of Columbia University in New York. They'll have debate teams come in, and usually it's things like former president versus a Secretary of State, and they're on the opposite sides of an issue, and they're bringing up really deep and clear points about all kinds of interesting topics. So I love it. I actually think that we should do versions of it here. Well, let's go ahead and dive into this. Let's have a prayer first before we get started here. Father in heaven, as we come together and talk about origins, we'd ask that you would clear our minds and help us to not only think clearly, but come into tune with your heart. And please lead us through this material as we go through it. We thank you for this. In Jesus' name, amen. All right, so I am recording, and so hopefully that you'll have a copy of this if you need it. And there's all the screens going, so you just choose whichever screen you want to look at, I guess. I haven't taught in here before, but that's kind of handy. I'm going to go pretty quick on some of this, because I'm assuming, if I understand correctly, you guys have all good science backgrounds, all essentially math science majors, except for you. You're like, great. Okay. Well, nonetheless, my intuition tells me that we probably will still be in good shape if we go through it quickly. Here's my outline. Very brief review of genetics, talking about, importantly then, what's a kind of the genetic mechanism of microevolution, and then switch right over and talk about evolution in the context of biomedical research. That's my background. And then lastly, wrap up with looking at chromosome 2, comparison between humans and great apes, within a small debate, what a surprise, at the end here, where you guys can kind of wrestle with some of that information and see what you think. All right, so first off, some principles of genetics. As you're well aware, all life is made up of cells, right? So cells make up all life, and inside of each cell, there is a nucleus, at least in eukaryotic cells, and in that nucleus is DNA, of course, DNA containing the instructions for how to put together those cells and all of their machinery. DNA, as you're again well aware, encodes for primarily just proteins, although there's some ribonucleic acid instructions, of course, in there as well. But the end goal is to take that DNA, transcribe it into RNA, and then eventually translate it into protein. And it's that protein chain that's actually the molecular machine that the cell needs. So you have roughly in your own human genome around 20,000 genes, and each of those genes can encode for at least one, not more, molecular machine. And the reason why I call it a molecular machine is because it quite literally is a molecular machine. This is where I wanted to switch over to a YouTube video. Before I do, what this picture I'm about to show you is looking at dynein molecules, which are these little motor proteins. They carry vesicles or really anything that's attached to them, and they walk along long tubes of, they're actually called tubulin. These microtubules are roads. They're essentially highways. And inside of cells, you have highways, even in, this is a neuron where there's a giant axon coming out of it. And of course, you know that motor neurons, for example, you have 3-foot axons, and you can imagine how you need highways if you're only producing proteins in the nucleus, or only producing proteins in the cell body, and you need equipment and materials and all kinds of proteins shipped down. You've got to have highways to do that. You have to have machinery to get that done. The video here is from a laboratory that was associated with MIT that their goal was to create as realistic 3D animations as possible of things that are at the sub-microscopic level. That's their goal. So let's take a look here and see what that looks like, just to reemphasize. So switching over all 3 TVs. And here we go. Let's see what happens. Oh, yeah. Whoa! Dramatic music. So those are just microtubules. They can disaggregate or aggregate to form the highways wherever the cell needs them. So you literally build the roads where it needs to go, and you can take came down. That is exactly what it would look like if you were able to get small enough and watch the dying molecule. It's often described as a drunken sailor as it walks down the highway. To get it to stop, quite literally what happens is, in fact, I'll back and pause. One summer I was at a conference that was just exclusively about axons, and of course these molecules are incredibly important to help move material to axons. And they were talking about, well, how do these actually work? In other words, this is all physical machinery. How does it stop? It's got to go somewhere, and when it gets there, what tells this thing to stop? Well, what actually ends up being are there are proteins that will wrap themselves around the microtubules and literally form a barrier that's not just a barrier, but it's got a little edge to it, and it'll pop the feed off, and when it does, it falls off, and subsequently the vessel is released at the target location, and this dying molecule will also then fall off of it. My whole point with showing you this is to make it very clear, just one simple principle, and that is, underlying all biology is this central dogma that says DNA transcribed into protein, transcribed into RNA, translated into protein, creates molecular machines, and each of these machines has one or more jobs that it's doing inside of the cell. Okay, that's the fundamental basis of everything we're going to talk about today. Questions at this point? Okay, back to this. All right. So, let's talk about microevolution. Have you guys had an introduction to microevolution at this point, macro and micro? I want to talk about the genetics underlying microevolution. It's a fairly simple concept, and that is this concept of when DNA is copied, each time it's copied, there's a chance that an error could be introduced. So a nucleotide, and forgive me for the very poor quality here, but what you hopefully see is these are all identical nucleotides everywhere, except for this base pair right here has been swapped out. There's an A here instead of a G, and then a T here instead of a C. And this happens whenever any organism replicates itself, it'll introduce errors. Now the rate of error is different for different kinds of organisms. Okay, in fact, we'll talk about that here in a second. But it's this phenomena that leads to the result of microevolution, that random mutations occur. Now, most often, these mutations are bad. Most often, these mutations are fundamentally bad. I'll give you an example of one, and you can argue with me whether this is actually bad or not, given risk for HIV maybe being reduced for someone who's hemizygous for this. Well, okay, fine, okay, I hear you. But switching back to just a simple principle, and that is, looking at hemoglobin, the beta chain can have a single nucleotide mutation, and as a result, that single nucleotide changes a codon, and you end up with now having a different amino acid instead of what was perhaps supposed to be there, and that causes some structural defects in this machine, and sure enough, that structural defect makes it polymerize into long rods and changes the blood cells even in their shape, makes them very weak as they try to squeeze through small blood vessels. They can rupture, and if you're homozygous for this trait in humans, you end up having sickle cell anemia. Okay, so that's most mutations, if you just introduce a random mutation, most often they'll end up in this category. Now, actually I have another example just to make it very clear. Retinoblastoma, the retinoblastoma gene, RB, is one of the first very clearly known oncogenes, and this gene basically helps control the cell cycle, and what the phenomenon is, if you have mutations in this gene, is you end up with retinoblastoma, which is cancer of the eye. The retina develops a tumor, and what you're seeing here is this very abnormal kind of reflection of the flash of the camera, and that's because there's a tumor in the back of the child's eye. Very commonly just in one eye, and the treatment is to remove the eye to prevent the tumor from spreading. Again, an example of mutations that are detrimental if you have them. But are there any beneficial mutations? Well, I would argue pretty strongly, at least on the microevolutionary scale, that yes, we can find examples where there are changes that increase the likelihood of survival or the ability to reproduce in some organisms on the short time frame. Let me give you at least one example, and that is HIV. In fact, that's what I was on. This is the reverse transcriptase of HIV. This is the molecule that allows, there's only 3 genes in HIV. There's a gene for the envelope, a protease gene, and then there's a gene that allows for it to copy itself, its own nucleic acid, and this is the reverse transcriptase doing that copying work. Well, if you look at the mutation rate between, we'll try to back to these sort of, I'm looking at Richard because we used to fight these sort of errors whenever we would, we're back in the class. All right, so in humans, mutation rate is very low. You have roughly 6 billion base pairs. That's because you got about 3 billion from your mom, about 3 billion from your dad, and you, on average, only have about 1 mutation every 35 million base pairs per generation. So in the production of gametes, either egg or sperm, only 1 error every 35 million nucleotides. That's pretty amazing, actually. That's because there's a lot of repair mechanisms built into your DNA that's very careful, that double checks everything, and even if it finds errors, can actually correct them the majority of the time. That means there's only about 175 mutations per generation. That may still sound like a lot, but it's actually not, because there's lots of areas of DNA where if you change it, it doesn't really make a difference on the phenotype. In other words, there's lots of intergenic regions of your DNA where you could plop in a change, no big deal, okay? If it happens to land in certain locations, very, very big deal, but nonetheless, about 175 mutations per generation. HIV is a little different. It's only 10,000 base pairs, the entire genome, that's it. 10,000. I remember the lab next to mine at the University of Missouri, actually both labs on either side were HIV labs, and on the wall of one of them was a big piece of paper about yay big, about yay tall, and it just had the entire sequence of the HIV genome just right there with annotations, of course, indicating which genes. So it's kind of interesting to be able to see the entire genome, every letter for a particular organism. Well, HIV is terrible at copying itself in one sense. That's because it mutates itself at least 1 mutation about every 10,000 base pairs. That means every time it tries to copy itself, it messes up. Probably at least once. There's a change. In an active infection in a human, about 10 billion viruses are made daily. Okay, that's a lot of viruses. What does that mean? It means every possible single and double point mutation every single day is generated. What does that mean? Well, if you think about the 10,000 base pairs, I'm just going to start to write them out here, right? So, and just going on forever, right? Going on for 10,000 base pairs. A single base pair mutation of this would be, well, we change the first letter. So the first letter becomes a T, and then everything else is the same for the rest of the 10,000. Okay? And then we would do it the next letter here. So let's do a G. And so there'd be whole viruses that they're each just 1 letter apart, 1 nucleotide different than each other. But not only every single point, so that's every single base pair could cycle through every single possibility, but every double. And that means the first 2 and the next 2 and next 2, and then separating them out, the first one and the third one, the first one and the fourth one, the first one and the fifth one, with every possible combination. That's impressive, right? So what do you think happens then when a person with HIV, they have an active infection, you give them a drug, and the drug fits in a certain physical location inside of perhaps the reverse transcriptase, there's reverse transcriptase inhibitors, fits in there, and as it fits in there, it blocks it from working. Well, every day there are variations of this virus that produce slightly different shapes of this molecule, and it's not a surprise at all that some of those shapes might be able to still function even when this drug is present. Maybe their shape is slightly different, so it kind of ejects the drug out of the active site, so it's not blocking it. And nonetheless, this is why HIV for so many years was so difficult to treat. You probably, I know you look at your face and see how old you are. You grew up in a generation which really didn't know how bad HIV was for so long, right? It was basically a death sentence for a very long time because every drug is simply within days or weeks, the virus would have adapted to it, and you would, of course, it would still continue the infection. So now, of course, they use cocktails. You use 2 or 3 drugs, and you use different ones that will trap the virus so it can't mutate to be able to defend against all 3 at once, if that makes sense. But nonetheless, the main point here is that microevolution is definitely occurring. If a drug is given and only one copy of the virus perhaps is resistant against it, but all the rest of them are susceptible, you can bet yourself that one copy that actually works is going to copy itself, and it's going to have a lot more progeny of that copy of the virus, and therefore natural selection has now selected for a copy of the virus that is resistant to that particular drug. So microevolution in a human body, essentially, with HIV occurring. It's a good classic example. Questions at this point? Yes, Ashley.
Well, I've been reading about like malaria recently
Yes.
and sickle cell anemia, and so I think it's interesting how how we've been able to like have drugs for malaria to fight against malaria, but then how the virus always develops resistance, and I think now if malaria was eradicated, but if somebody were to make it in the lab and then like start it all over again, then we'd be in trouble for sure. But then at the same time, sickle cell, if you are like heterozygous for sickle cell,
Right.
That virus, the malaria virus, has still not been able to like understand how to get around that. You become like significantly immune to malaria when you have sickle cell. Why hasn't the virus, or do you know why the virus hasn't like been able to understand this biological prevention, but it can like rapidly change function whenever it's presented with like a synthetic.
That's a good question. So first off, keep in mind malaria is a plasmodium. It's an amoeba, is not a virus, but that's okay. But it's still it's a parasitic little organism. I don't know if I can answer off the top of my head why sickle cell. Of course, it's not completely protective. If you are hemizygous for sickle cell trait, then you have reduced risk around malaria, but you can still get infected with malaria. Whereas there's not a lot. There are drugs, though, however, that do have a fair amount of effect on an active infection that's ongoing. There's just nothing that has quite been able to stop the infections quite in their tracks. It's not completely effective. Why is that? Drug research is hard.
Okay.
I'll put it that way. Yeah. I'll chalk it up to that for right now.
I just think it's interesting how like some of the biological mechanisms seem to be
Actually more effective in some ways.
Have you been reading Michael Behe's book? Is that?
Yeah.
He talks about that.
Yeah, that's interesting.
If HIV has that high of a level
Yes.
mutation rate,
Yes.
is it common for it to mutate itself, like
Oh,
not
yeah.
completely, but into like a non-effective?
Oh, that's the beauty of it. Yeah, it has gotten so many totally messed up forms every time it does it, and those will not replicate. They're all messed up. They can't replicate probably many times. The vast majority, in fact, I don't know, I shouldn't put a percentage on it, but I'll go ahead and throw one out there. Probably 99.99% of the mutations that are introduced hurt rather than help. But we're dealing with such big numbers. You get one good one, you can lose billions. It's okay. Yeah.
My memory is last time I heard you give this lecture, you
Yeah.
said there are really only 3 viable forms of HIV. Is that correct, or am I misremembering?
Well, there's only a handful of mutations that it can have that are compatible with replicating itself, because it's got to maintain certain structures. But there's definitely patterns to that. So one of the big things in HIV research is studying what are all of the versions of the HIV that are out there, and which versions are susceptible to which drugs. There's certainly more than 3. There's certainly more than 3, because each protein can have actually a whole bunch of changes and still get the basic job done that it's supposed to do. Anyhow, main point, microevolution is occurring, and it's because it can have mutations, and those mutations, vast majority of mutations, don't help, but maybe a few could help potentially. I want to talk for just a couple of minutes about evolution and the relationship to biomedical research, and I'm going to use a particular disease, spinal muscular atrophy, as an example, and this is the disease that I worked on for my graduate work. So first off, what is this disease, spinal muscular atrophy? It's frequently abbreviated SMA. It's a genetic disease, inherited. It affects children. It causes paralysis of voluntary muscle, so skeletal muscle. It gets progressively worse, the paralysis does, and then it most often leads to death. Most common form affects children, and their first signs are seen at about 6 months of age, and most commonly in that form they die before age 2. Stephen Hawking, as you guys probably have heard about in the news most recently, passing away. He had a version of a motor neuron disease, and this is a motor neuron disease. It's the same targeted cell that controls cells that control skeletal muscle, but the difference being the time of onset. So here's some examples of children born with SMA. The most severe type, which they most often die before age 2, is type 1. That's the most common, unfortunately. Type 2 is a little bit less severe. They can live into the almost adolescent years, and type 3 can actually have almost a normal lifespan because their paralysis is the most minimized. You can probably see this guy's arm, how thin it is. The skeletal muscle, it gets smaller because it's not being stimulated very much because the motor neurons are dying that's controlling it. What's causing the paralysis, I've hinted at it, it's the motor neurons in the spinal cord, specifically the lower motor neurons that are dying. And when they die, it breaks the connection between your brain and your voluntary muscles. Talking about genetics now, all SMA patients have mutations in a single gene, one gene. It's called survival motor neuron. They were very clever, right? Survival motor neuron. If you don't have this, your motor neurons don't survive. You need this gene. The genetics of this is very interesting. So if all of the patients have mutations in the same gene, why do they have this spectrum of severity? Some of them are severe, some of them are a little bit less severe, and some have very mild symptoms. Well, here's the interesting part about the genetics. The gene survival motor neuron SMN1 is in a region of chromosome 5 where there's a pretty large duplication. You might be surprised to hear this, but in the human genome, there are thousands of duplications. What do I mean by duplication? Quite literally, in this area of the DNA, there is a region that looks as though this part here is exactly the same as this part over here. If you start to read the genes off, notice H4F5T, that stands for telomere, towards the telomeric end of the chromosome. H4F5C, that's this side more towards the centromere, towards the middle of the chromosome. And then there's SMN1, there's SMN2, and so on. Each of these genes are almost identical copies, almost, but not quite. As an example, SMN1 to SMN2, these cover about 30,000 base pairs for each gene, and there are 5 differences that land within that coding sequence. So not very many differences, but there are a few differences. Okay. Well, it turns out that one of those differences in SMN2 makes it a weak version of the SMN1 gene. It still works, but it works at very low levels. So what happens in our patients? Turns out that in normal individuals, most likely in this room, you have healthy copies of both SMN1 and SMN2. Imagine if this was the maternal copy and this is the paternal copy. What ends up happening, oop, it skipped. Oh shoot, guys, I'm going to have to draw. What ends up happening is that in SMA patients, they lose their SMN1 copies due to usually deletions, meaning the DNA is simply not there. And they're left to rely on their SMN2 copies, which are weak. Each SMN2 copy produces a little bit of functional protein, and as a result, the more SMN2 copies you have, the less your symptoms are. And so we can actually very clearly explain why some kids are very severe and some are very mild. The most severe ones will only have 1 or 2 copies of SMN2. The more mild versions will have 3 or even 4 copies of SMN2. So leading you through that same journey, now you understand the genetics behind it. What do we do in the lab to study this? Well, note that genes are often very similar between species. This is the SMN1 gene, and we're comparing it between different species. At the top here, you see Homo sapiens twice. That's because Homo sapiens, this is the top one, is SMN1, and then this is SMN2. And you remember I said there were some slight differences? You can see there's no coding differences. It's identical all the way across. Below that, you see pig, and then dog, and then cow. Keep on going down the list. Do you guys even know some of these? It's okay if you don't. I'm not big into memorizing there, but you want to know what this one is? The musculus. That's the mouse. This one you should be able to pick up. Rats. How about this one? What's that? Chicken? Yeah, chicken. Yeah, chicken, Gallus. And then this is a zebrafish. These are the common research organisms that are used. But I want you to notice, this is the amino acid sequence. And if you look at that amino acid sequence, do you notice something there? Wow. It is like very, very similar. Like if you go down the list, I mean, this is a fish on the bottom, right? And still, there's a lot of similarities. If you look at the human to the pig, whew, man, there's only a few differences that you might be able to pick up on. Same thing even with the mouse. So that's one of the first principles I want to highlight, and that is in biomedical research, one of the things that underlies it is that there are tremendous similarities at the genetic level between organisms, which lend itself then to testing out things, potential treatments, for example, in models, animal models. So 99% of human genes have an analogous partner in mice. Mice can be genetically engineered. And sure enough, SMA mice have been made. Mice don't normally get SMA, never. They have 1 copy of SMN1 and it works. And it's rare that it ever is deleted. Well, you can actually delete the mouse SMN and then put into the mouse SMN2 copies. And you can put in different numbers of copies and you can create different severities of the disease, if that makes sense. Now, why would we want to curse poor mice with a human disease? Again, it's so that you can test out potential therapies. Here is examples of litter mates at day, goodness, this is probably about day, I'm going to go with day 12 approximately. And you can see this guy's nice and healthy, and this guy right here is all paralyzed, essentially. He's well on his way towards dying. Unfortunately, he won't be able to eat. And that's because he's been genetically engineered to have the disease. So, I've already gone over that. So here's the major conclusions I want to pull out of that. By the way, let me end on a happy note. Can I end on a happy? Because you're like, oh my goodness, these people torturing these animals. Well, I certainly didn't enjoy torturing poor little helpless animals, but there's a lot of kids out there and a lot of families that are affected by this disease. And so we use this model as a way to test out therapies. And when I was leaving the lab during my postdoctoral year, we engineered a version of a virus, an adeno-associated virus, which carried a complete copy of the SMN1 gene. Does that make sense? You can actually take out the virus DNA, huge chunk of it, 90+% of it, and you put in the human SMN1 gene. The virus is pretty innocuous. 60% of the population already has adeno-associated virus infections that are replicating at very low levels, and they're not causing a symptomatic disease. And that particular virus was able to infect early on, at least in the disease, like when you give it to them on day 1, we could, of course, diagnose which ones are going to have the disease very early, and then inject the virus into them. With normal drug treatments, so talking about drugs, the furthest old mice we could ever get was about 23 days, and that's bumping them from an average of 17 days. The first virus-treated mice that we used, they were living past 100 days because it was repairing the paralysis to that extent. And researchers at the University of Ohio perfected that particular technique, and they have a company, Avexis is what they call the company. It's now worth several $100 million because they now have into phase 2 clinical trials in humans, taking a version of the virus and treating kids who have this disease, and thereby preventing them from not only dying, but maybe giving them back substantial motor function. Just a little side note there to make it not so depressing about working on those poor little animals. All right. Questions there? Questions there on that section?
So
Yes.
you use both of your SMN2 genes at once, always, or?
Oh, your SMN1s? Yeah, the
Yes.
SMN1,
Sorry,
the SMN1s
yes.
that if you
Yeah.
were to lose them, it would cause the disease.
Yeah.
What often happens is no, there can be people who are carriers. About 1 in 40 people is a carrier, and they only have one copy of SMN1, but they're perfectly fine. You would never know it. They could be an athlete, could be everything. They don't lose any motor neurons. They still have they have sufficient amount of SMN protein being produced. It's when two carriers have a child that produces a child that has no copies of the...
Just missing from their genome.
It's missing from their genome, exactly. Yeah. Yeah. Good question. Yes, Richard, question, yes.
Mostly because this is something my family has. Do you know anything about the alpha-1 antitrypsin?
Deficiencies, yes. Mm-hmm.
Is it a similar type of thing where, there's two genes involved? I'm PISS, if anyone cares. My mom's PIZS, which is much worse. My mom's cousin is PIZZ. Actually, I think this week is having the surgery for her liver
Transplant. Is it liver? Or it should be lung, right?
Well, it affects, I think it's lung and liver and kidney.
Right.
It's either liver or kidney she's having transplant.
Oh, my.
So
Right.
I'm very curious about, there's two genes, which is why there's an M as well, but that would be fine and normal. We don't have that in my family. But are the genes similar in, you have two different genes we were talking about there, the SMP1
Right.
and SMP2.
Mm-hmm. So when I said I knew about it, I knew about as much as you just walked us through. my PhD's on this one, not that one there.
Okay.
Yeah. Just to bring everybody up to speed, alpha-1 antitrypsin is an important protease inhibitor. Proteases are enzymes that digest protein, of course, and normally, of course, we think about proteases like in our digestive tract, and they eat up protein to help us digest food. That's good. There's also actually proteases in your lungs. That's one of the affected organs in people who have deficiency in this enzyme. And those proteases are normally in a balance where there's a little bit of protease activity present, which is a healthy, normal thing in assisting in breaking down particles and things that might end up being in your lungs. But there's also a fair amount of anti-protease activity to block it from getting too aggressive. Because if it gets too aggressive, it starts to break down normal tissue. And you can get emphysema. People who never smoke their entire lives can get emphysema because they have genetic deficits in the particular enzyme you're talking about that blocks proteases. And I think you're absolutely right. There's other effects for other organs too. I don't know beyond that, unfortunately, Richard. Questions? Other questions about anything that we've talked about? Well, I want to lead you through this one because this is what the debate is going to be about here. Human versus chimpanzee chromosome 2. Okay, first off, let's review how DNA is kind of stored in our cells. Most of the time when a cell is not actively dividing, it's stored in a very loose form of a kind of loose chromatin. But when it's going to divide, it condenses, right? It condenses in these very clear condensed chromosomes, okay? And that's, of course, the kind of showing what it looks like there. Here's an actual scanning electron micrograph of chromosomes that had come together probably right at metaphase and then were captured for this particular image. Here's another image of them. This particular image is just showing that you can paint chromosomes with labels. So, for example, based on the sequence of the chromosome, you can create little colored probes that have a dye on the back of them, and then on the front end of them, they have little adapters that match the DNA sequence in that chromosome, so it effectively will always bind to and label that chromosome, and you can make it bind to different pieces of that chromosome, so you can effectively paint the whole chromosome with a color. So when you look at it, you can quickly look underneath the microscope and actually look for, oh yeah, here's all these different chromosomes. I know which chromosome it is based on the color. And then also you can even detect when chromosomes abnormally recombine. That can happen where there is abnormal crossover between chromosomes that are not homologous. So maybe between chromosome 1 and chromosome 5, and they'll swap pieces. Sometimes it's okay. You can get by with it because maybe it doesn't destroy any DNA information, but often it will really mess up important genes or important gene structure and then results in non-viable embryos or diseases. Well, what about chromosomes? One thing that's worth noting, there's a little middle portion, the centromere on each chromosome, that allows it to stick to its homologous pair. Recall that for each chromosome, one half, so like let's say this half would be maternal side and this side could be, in this particular situation, this would be in the duplication of this chromosome, there'd be two identical halves. Both of them might be from maternal or both of them might be from paternal, if that makes sense, right? And they've just been copied and you're getting ready to maybe pull them apart. So the centromere is in the middle, and then there's a telomere on the end. Telomeres are specific sequence of DNA that's on the end of the chromosomes, which is there for a particular function, and that's when you're copying DNA, when you get to the end, the polymerase is kind of big. It's like a big giant machine, and that big polymerase can't quite copy all the way to the end. It always kind of finishes prematurely because it just can't physically fit on the end of the DNA, and as a result, every time it copies the DNA, the end of the DNA gets shorter. Well, that's no good, right? Because you start eating into DNA, and between generations of dividing cells, you would just like whittle down the DNA of the chromosome. So the telomeres add on essentially buffer DNA that can be added on in between cycles, and that buffer DNA, no problem. You can lose some of it, and you're not really losing anything of importance, and if necessary, it can be re-added through an enzyme. Well, you can paint those things. This is painting the telomeres on the ends, those little bright ends of the chromosomes there. Those are telomeres showing up with labels. You can also paint chromosomes. This is like a Giemsa stain of a chromosome 5. This is where the SMN gene is. It's specifically right there. 5q13.1 is right this journal location. This represents the entire chromosome, 180 million base pairs, about 1,000 genes sandwiched into that chromosome. Well, back to the point about chromosomes and comparing them to great apes, how many chromosomes do humans have? Again, remind me, how many do we have? How many?
23. I'll be real tired.
I'll be retired. Hey, it's okay. No penalties for in-class answers. No penalties for in-class answers. Yeah, there's 23 pairs. If we count them up, we can actually see them up here. So here's chromosome 1, 2, and then all the way down to X and Y. But then you multiply. The reason he got the 6, I'm almost sure if I could go into his brain patterns, is because when you take the two copies of it, you get 46, right? So 23 pairs, a total of 46. They're numbered in a specific order, and that's because the biggest one is chromosome 1, the next biggest is chromosome 2, chromosome 3. It's just based on length. So if you get down here, you can just kind of see how they get smaller and smaller and smaller all the way through the end. Well, something interesting is out there is that the chromosomes, there's different numbers for different organisms, and the number does not reflect the complexity of the organism at all. You can have, organism like a dog has 78 chromosomes. Doesn't mean that they're more complex. It just means that their DNA is chopped up into more pieces. If that makes sense. They don't have more DNA. In fact, actually, they have about the exact same amount, total amount of DNA as humans. But of course, the instructions encoded are different. All right. So here is the conundrum. The question at hand. What happens when you compare chimpanzee, or any great ape, so chimpanzee, gorilla, orangutan, these are all great apes. When you compare their DNA to our DNA, they have 48 chromosomes total compared to our 46. Interesting. Basically what it means is they have one more chromosome pair. And so what if you can, we know there's a lot of similarities between the way that they look and the way that we look, and at the DNA level, if you compare their DNA level, it's very similar. How is it laid out structurally? Well, if you just look into these, you'll see the H is the human, the C is the chimpanzee, this is chromosome 11, this is a different kind of banding pattern. I'm sorry, I got such low quality pictures, but you can still tell there's just a lot of similarities in that banding pattern. That's underlying the similarity of sequence for that particular chromosome as you compare them. Okay? Well, if that's the case, then where's this extra chromosome? Do they have like an extra chromosome that does something completely different than ours? Not exactly. Let's come up to chromosome number 2 and take a look and see what's going on there. Look at chromosome number 2. Here's a human one, right, going all the way down. Chimpanzees and other great apes have what they call chromosome 2A, and then down here, they put it down lower, 2B. Do you notice something there? It appears as though human chromosome 2 kind of covers the same genetic information as chimpanzee chromosome 2A and chimpanzee chromosome 2B. Okay? Let's take a different look here. Here's, again, just chromosome 2, comparing between the different species there. And again, this is a giemsa stain, and the giemsa stain underlies DNA structure, and sure enough, the banding pattern, as it lines up, what you see is essentially that. Okay? Well, what in the world could be going on? Well, here's where it gets interesting. In the middle of human chromosome 2, we would expect there to be, of course, a centromere. And there most certainly is a centromere, but look where the centromere is. It's actually right up here. It's this orange little dot in representation. Further down the chromosome, there's this region right in the middle where there is this sequence of DNA that looks very much like telomere sequence, but not quite precisely telomere sequence. And telomere sequence on our ends is T, T, A, G, 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 repeated again and again and again. Okay? Now, if you look in there, what you'll see is, you'd be like, oh yeah, I find one of those. Like, here's one right here. Okay? But right below it, like that one's not quite right. TT, and then it's got 4 Gs. And this one's kind of a whole bunch of Gs there. And this one's quite off a little bit. But nonetheless, there's a general pattern that kind of looks a little bit like a telomere. Does that make sense? And then actually the bottom part here, the C's there, this is actually just reading the other direction. If you translate this, if you give what the opposite base pair would be, if you're going kind of reading it in the opposite direction, you would see the same thing. Let's say this guy right here. Well, actually, no, this guy right here. The A translates to a T, the A translates to a T, the T translates to an A, and then you got 3 Gs there. The C's all go to G's. If you read it from right to left, it basically looks like a telomere sequence going in the opposite direction. So the scientific hypothesis behind this is that at some point, and again, speaking from an evolutionary perspective here, and explaining that, and I'm going from there, the 2 ends of 2 chromosomes, which were present in some common ancestor of humans and apes, on the human lineage, 2 of those chromosomes fused together. And their 2 telomeres fused, and then subsequently the DNA sequence has degraded because it's no longer needed and being used. And there is an extra centromere sequence. There's centromeric sequence as well. I'm not going to show that. But that centromeric sequence, there's like a rough version of that that's sitting there. So, interesting. Here's what I'd like for you to do in the next couple of minutes. In small groups of 3 here, I suppose, I want you to compare and contrast a creationist model to understanding that information versus an evolutionary model. And then I'm going to ask you a few questions. So you better quickly put your heads together and let's see what we come up with.
There's always the uncontrolled, like, things that function in small ways, they like run in the same way. These basic functions, so the things that are like the genetics for those creatures that don't have these things, they need
Short is basically they seem to be working on issues which can't take this.
Yeah, and how document all this stuff, like, just because
Looks
it's
close,
doesn't,
but like.
doesn't necessarily mean that it came from something similar, but that was created by one
Yeah.
person, you know?
Right, so it doesn't mean that a God couldn't have made things just similar. Like, that's just a little more of the
It just depends on what conclusions you draw.
Yeah, I mean,
It's
it's
not
not
that
that
they
Like, is
You
it
can
just,
definitely see the evolution.
is the
of
similarity
creation.
of their such rare creation is the end of it?
I mean, I
Or
think
should
that-
they look more at what the functions are and maybe that telomere region that
Because it's an outlandish.
taken out of everything, but over time,
It's
was
kind
because
of
it was not functional. But then again, here,
I don't know,
you can't
maybe-
backtrack and say what
Oh,
are telomeres.
with like philosophy is
We
like
can say what it should have
type
had,
of
it
vision
shouldn't
that
have,
can be
but we
extrapolated,
can't
I
predict
guess.
that's what was in
I don't
that
know.
sequence of the.
Oh,
Yeah,
just
and
like
there's
We're
that,
assuming
there's
it
kind
got
of
taken out.
bias,
We
in a
don't
way,
make
just
some
on
assumptions.
kind of like what you want to happen, you know? Like you had this opinion and you want that, that's just an opinion.
I apologize to interrupt, but we do have a short amount of time, so I want to hear what you guys have to share. If you look at the sort of raw data and information, how would we look at that from, and we'll start with perhaps the easier one, the evolutionary perspective, and then I'd love to hear also the creation perspective. Let's start with the evolutionary. Do you want to give a venture on like what that perspective is? Of course, I gave it. I shouldn't have just, like, dumped it in front of you there, but I guess I did. You know what, or anybody else?
Oh, sure. Just, I mean, just that we, at one point, there was a mutation, chromosomes
Right.
combined, and we actually did come from, this could be used as evidence that we did evolve from primates or.
And a common ancestor with primates
Yeah.
that are today. Right. Yeah. Okay. How would a creationist model interpret this raw data and information? What do you guys think? Ashley?
Well, I don't know if you can really draw a conclusion from this as a
Right.
creationist, but I do think that just because the DNA is similar, you have to think like the biological functions are kind of similar. What we have to do, like our bodies, like our mass is like similar. We have to use our arms and legs in the same way. So like a God could just like, oh well, they're gonna need this. This is how this organism is gonna need to have like this DNA in order for their functioning to work. So it's possible that a God just made things similar because that was the best way for their like enzymes or proteins to function for that purpose.
Interesting. So there could be some functionality to it, is what you're trying to say? Maybe
Yeah.
there's function to the way that it looks fused, but maybe there's a reason behind it.
Yeah.
Okay. That's possible. What other thoughts do we have here?
I mean,
What about time frame? So is it possible that in a relatively recent time frame, we know, of course, that new mutations occur on a relatively frequent basis, and not just new mutations, but new recombinations of chromosomes? Could it be possible that a relatively recent ancestor of human lineage that all of us have, so we're talking a Noah-like figure, could have had a recombination of two chromosomes? Maybe he was originally created with 48, as other great apes were, and then there was a recent recombination. We don't know exactly in time if that event occurred, when did that occur? Does that make sense? We can't pinpoint, we can't say for sure.
so you're saying like even if in fact it did occur from an ancestor where they were fused, that would not necessarily
Exclude.
a creationist viewpoint?
That's what some would say, absolutely, because we know that there are lots of diseases and mutations that do pop up on a single generation basis. So it is possible that that could have occurred and been inherited, passed on. Nonetheless, it is a pretty confounding example of the relationship between genetics and evolution and creation and looking at origins. And here's the final thoughts I'd like to leave you with. I have just a couple, and then you can run out the door here. And that is, of course, we know what DNA does. It encodes information for those molecular machines, proteins. We know that mutations arise in DNA. Most of them are harmful. Some mutations are, however, beneficial. Genes are often similar between species, and this does fit either a macroevolutionary perspective or a creationist perspective. Animal models of human disease are a result of having that similarity. And then again, with chromosome 2, in humans versus chimps, it does fit either. It really boils down to a perspective of a common creator or a perspective of a common ancestor is what it boils fundamentally down to. And my last thought for you, and it's an important one, I would hope that each of you would take out of here. As I've gone through my additional years of training and working with a lot of really smart people, one of the things that I would find helpful if I was in your shoes today as I was going on the rest of my career, and that is, as you look at evolution and you look at creation, unfortunately perhaps, or maybe not, there is not just a slam dunk information that's out there. And as you work with people with diverse views, I would encourage you to always keep an open mind with those individuals and to not try to force down someone's throat one perspective or another, because it is very complex as you look at it. And you will run across very honest, very intellectually honest individuals who view this in different ways. Kind of the same way politics is, actually, in some ways. Although those have gotten extremely polarized, perhaps, extremely polarized. Boy, I wish I had more time with you guys. I'm sorry, but it's been fun. If you want to have more questions for me, I'll stick around for a few minutes, but I know many of you probably have other classes.
Unfortunately, we have a genetics test.
Do you?